So here's some of the key vocabulary in this story of cellular or aerobic respiration. Oxidation, and reduction. Two terms that I want you to be able to define and know what they mean. Oxidation is the removal of electrons. That's oxidation. You're breaking things down. You're tearing them apart. So, oxidation, removal of electrons. Reduction is the opposite. It's the addition of electrons. So you're building things up by adding to them. Now, oxidation and reduction are always coupled. You never have free electrons just wandering around willy-nilly in the cell. They're always bound to something. So let's say these are always coupled. So if one molecule is oxidized, having lost electrons, something else has to be reduced, gaining those electrons. So if one thing's reduced, something else is going to be oxidized. They're always coupled together. And that chapter of chemistry deals with redox reactions, short for reduction oxidation reactions. An example of oxidation and reduction. Now, even though we're being filmed, if I erase too fast, just holler. Okay? I'll try to give you a couple more seconds. An example of oxidation and reduction is going to involve these electron carrier, or this electron carrier, Nadh plus and NADH. And they switch back and forth. It's the same molecule. It switches back and forth between the oxidized and the reduced state. So let's say, identify this as our primary electron carrier. So NAD is oxidized. It's missing what? Yeah, 2 electrons. And these are high energy electrons from the glucose. Remember that the energy in glucose is in the bonds, and the bonds are made up of electrons. So some of those electrons are high energy. Remember, electrons can exist in high or low energy states. So we're capturing high energy electrons. This is the form of NAD when it's reduced. So what's happened to it? It has gained 2 electrons. So make sure that you're clear on the definition of oxidation and reduction in this sense. The removal or the addition of electrons. Okay. So, introducing the summary or the overall equation for aerobic or respiration. This I want you to know. So we start out with glucose, which is a 6-carbon sugar. So we'll say C6H12O6. We could write glucose if we wanted. plus oxygen. Those two, sugar and oxygen, will yield, upon completion of the process, CO2 and water and energy. And if you want to balance the equation, it's 1 sugar and 6 of everything else. So it'll be 1 sugar and 6 O2 gives 6 CO2 and 6 water. So when you balance an equation, you'll do this in chemistry, not here in biology, but all of these atoms on this side of the equation have to be accounted for in the other side of the equation. So you can play around with it and see if all of these are found on the other side as well. The energy that we get out of this, the whole process is aimed at capturing the energy from glucose as we break it down step by step, capturing that energy. At the end, the energy is contained in ATP, and that accounts for about 40% of the energy in glucose at the end of this process is captured in ATP. And the rest of the energy is released as heat, about 60%. It's rather inefficient, but as far as chemical reactions go, that's pretty good. Your car may get, depending on what you drive, maybe 12% of the energy from the gasoline is used to make the car go down the road. The rest of it goes out in heat and is lost as friction. So this is pretty efficient. Doesn't look like much, but 40% gain is pretty good. This should look like one of those 2 laws of thermodynamics that we went through yesterday. This loss of energy and the formation of heat. Check your notes. See if this fits with the second law of thermodynamics, which I'll abbreviate as TD. Okay? So this process of aerobic or cellular respiration that we have summarized in this equation gets done in 4 steps. Done in 4 main steps. And they are, the first one is glycolysis. And glycolysis happens, it occurs in the cytoplasm of the cell. In other words, outside of the mitochondria. The other 3 steps, 2, 3, and 4, they all occur within the mitochondria. So the second step, after glycolysis, is sometimes referred to as a prep step, where you are producing, you're converting pyruvate to acetyl coenzyme A. So let's say pyruvate to acetyl coenzyme A. That's the second step. And the third step is the Krebs cycle. That's the term your book uses. Other books refer to the Krebs cycle as the citric acid cycle. Sometimes other books, not so often, refer to it as the TCA. They're all the same thing. Your book uses Krebs cycle, that works for me. That's the third step. We'll go through these in order. The last step is the electron transport chain. Or the ETS, for electron transport system. And again, these all occur in the mitochondria. Abbreviate mitochondria. Okay? So we'll erase that, and then we'll go through the first one with glycolysis. Okay, step one, glycolysis. So we are going to be splitting glucose in two. So let's start with glucose. And through a series of reactions, which we are just going to summarize here, but through a whole sequence of reactions and enzymes and intermediates along the way, glucose is split in two and produces two pyruvate molecules. That's the end result. So glucose in, 2 pyruvate out. I like to keep track of the carbon. I think it helps as you go through all of these 4 steps. So glucose has 6 carbon, and pyruvate has 3 carbons each. So I'll just write it as 3C. But there's 2 of them, so we still have our 6 carbons. No carbon gain, no carbon loss. Try to keep track of those as we go through this. Also, try to keep track of where the energy is. This is a story about the transfer of energy, ultimately from the sun in the plants to make the sugar, and then for us and other animals, we take the sugar and we break that down and get that energy back out. So try to look at this, follow the carbon, and try to follow the energy. One thing that happens that we have to include in the summary of glycolysis is that this process requires an input of energy. So we have to invest 2 ATP to get glycolysis started. So those 2 ATP participate in these series of reactions that are just summarized. And at the end of glycolysis, at the end of these reactions, we get out 4 ATP. That's the out, that's our sort of our dividend. That was the input, that's the output. So overall, the net gain is only 2 ATP. So you don't get a lot of ATP out of glycolysis. You get some, but not very much. Where's the energy? Glucose is gone. The energy now, there's a little bit in the ATP. Where's the rest of the energy? Some was lost as heat, but on this picture, the board, where's the energy? It's in the pyruvate. Yeah, there's still a lot of chemical bonds to be broken down in the pyruvate. Now the other piece that we'll introduce here, we have to collect electrons. we're collecting those high energy electrons to send to the very last step, which was the ETS, the electron transport chain, which is where we're going to make most of the ATP. So we need to bring in the electron carriers, the electron bus. So what was our electron carrier? It was NAD or NADH. Which one was oxidized? NAD plus. So there is our NAD plus. I hope that shows up on the film. And that's the bus. It's empty. It's oxidized. It comes in and it picks up the high energy electrons and it leaves reduced as NADH. So again, what you should recognize is that this is oxidized. The NAD plus is oxidized. This is reduced. And what's important to remember is that it's carrying 2 high energy electrons. Okay? It turns out that we have to put 2 of these in and we get 2 of them out per glucose. So what do we get at the end of glycolysis? We get pyruvate, 2 reduced electron carriers, and 2 molecules of ATP. That's the end of glycolysis, how we're going to present it. Okay? So you might keep in mind, along with following the carbons and the flow of energy, you might keep in mind when you're trying to draw this on the dinner napkin tonight with your ballpoint pen, to try to remember how this works, think about what goes in and what comes out. What did you have to invest in glycolysis to get it to go? And then what did you get out of it? Well, you had to invest in glucose. That entered these reactions. 2 NAD entered, and 2 ATP. So there are your inputs, right? That's what's going into all of this. What comes out are the 2 pyruvate, the 2 NADHs carrying the high energy electrons, and 4 ATP. Right? So try to do that with each of these steps. Keeping track of the carbons, the energy, and then what goes in, which is above my hand, and what goes out, which is below. Okay? So there's glycolysis. And the second step is this prep step. What were we doing there again? We're converting the pyruvate that we just produced in glycolysis. We're going to convert that pyruvate to acetyl-CoA. So let's do it this way. Here is the inner mitochondrial membrane. So inner mito membrane, or the inner mitochondrial membrane. Remember, it has 2 membranes, an outer and an inner. This is just the inner one, and we'll put in a membrane carrier. This should look familiar from what we've done previously, representing a protein channel to that inner membrane. Pyruvate. Where is the pyruvate at the end of glycolysis? Where in the cell? It's in the cytoplasm, inside the cell, not in the mitochondria. So this is floating around out in the cytoplasm. And the pyruvate is transported across that inner mitochondrial membrane. And at the end, it gets converted to a 2-carbon acetyl group. And that acetyl group, which contains 2 carbons, how many carbons do you think are in acetic acid? 2 from the acetyl. So there's the 2-carbon acetyl group, and it gets connected to a coenzyme, coenzyme A. It looks a little bit like NAD. So that's the input, there's the output. How many carbons were in pyruvate? 3. So we lost 1. So one of them comes off as CO2. So we've lost one of our carbons, and that was showed up in that summary reaction, right? Where we started with glucose plus oxygen, yielded CO2 and water and energy. There's one of the CO2s. To get this to happen, we also bring in the electron bus and scoop up a couple of high energy electrons, and that leaves as NADH. So again, every time you see NADH, you are thinking, it's carrying 2 high energy electrons. And where does all of the NADH, where do all of these loaded electron buses go? They all go to the ETS at the end of the story. Okay? That is our Krebs step, converting pyruvate to acetyl coenzyme A. Okay? Drops off a carbon as CO2, and we scoop up some high energy electrons. Now we're left with acetyl coenzyme A, and that will be the first thing into step 3, which is the Krebs cycle. Where is the energy up here if we're following it? A little energy in CO2. There's energy here, right? Because it's carrying the high energy electron bonds. Well, there needs to be energy here, or why proceed any further? Okay, step 3 was Krebs cycle. So let's start at the top. We'll make it sort of in a circular fashion. So here is the acetyl coenzyme A. 2 carbons in the acetyl group, right? We're keeping track of them. So per molecule of glucose, how many pyruvate did we get? 2. Per molecule of glucose, how many acetyl-CoAs are we getting? 2. Okay, so this would be duplicated a second time for the other half of the original glucose. And so acetyl-CoA comes in, it binds to a 4-carbon intermediate. I don't care really if you remember what that is. It's discussed in your book. But it's for the first product of the Krebs cycle is citrate, which when that ionizes, it ionizes to citric acid, hence the name, the citric acid cycle, because we keep making the citric acid. This is a 6-carbon compound. I'm not going to do any other intermediates along the way. It's just the citrate. Now, we're done with the coenzyme. The coenzyme A is a shuttle. It's a carrier. Its job has been fulfilled, if you will. So the CoA splits off. It leaves. It's done. Where does the CoA go? Back to the second step to pick up another acetyl group that's being made from the pyruvate. So these things cycle in the cell. So there's the CoA. We're done with it. It's finished its job. And then through a series of reactions and intermediates, all catalyzed by different enzymes, we wind back up with what we started with. This 4-carbon intermediate, which will gain another acetyl group, making more citrate, and this cycle keeps running round and round and round as long as we have enough reagents, enough inputs to keep it going. So now we have to account for our carbons, and one of the first things that happens is that another carbon splits off and leaves as a CO2. And along the way, we bring in another NAD. Is this oxidized or reduced? Oxidized, good. It's going to come in and it's going to pick up 2 more high energy electrons and leave in the reduced form NADH. That again is going off to the ETS. And we then spin off another CO2. And we bring in another NAD plus and reduce that to NADH. Again, all those NADHs are carrying high energy electrons, 2 each. Then we keep going around, sort of grinding up the molecules that are left. And out of this, we can do a couple of steps. We can phosphorylate 1 molecule of ADP back to ATP. That's the only ATP that we get out of the Krebs cycle, is that one. And then we keep going around and we introduce a slightly different electron carrier. We've been using NAD, and that's the electron bus. There's another one, FAD. This is sort of like the Volkswagen bus, maybe. No plus charge on it. It scoops up a couple of remaining electrons that are not at quite as high of an energy level, but that's okay. It leaves, after picking up 2 electrons, as FADH2. FAD does not have a charge on it. When you put 2 electrons on it, it will have 2 negative charges. So we'll add 2 protons, cancel those out, and There's our FADH2. The point here is that it also is an electron carrier. It's carrying two relatively high energy electrons that are also going to go to the ETS. Just like all of the NADHs. And finally, we bring in one more NAD, because you put the plus charge on all your NADs. And then it leaves, again, as reduced NADH. Off to the ETS with two high energy electrons. So, if we're tracking our carbons, we entered with 2 from the original 2 that are left from the glucose, and they went out here and here. And remember, this whole thing is repeated for the other half of the original glucose, the other pyruvate, the other acetyl-CoA. So let's try to list our inputs. So for the Krebs cycle, what do you need to have going into it? You need acetyl-CoA, and you need this intermediate. We'll leave that alone. And you need NAD. How many NADs do we have up here on the board? 3. So we need 3 NADs. And what else? We need 1 FAD going in, and we have an ADP and a P going in. Then what do we get out? Well, you get out the 3 NADHs, the FADH2, both of those being reduced, and we get out this one lone ATP. And we get out carbon dioxide. So where is the energy now? We tracked the carbons, we figured out inputs and outputs. Where is the energy? Well, that's just one. We're not getting a lot out. We got more out in glycolysis. Yeah, there's some ATP or some energy there, but where's the bulk of it? Yeah, it's in those high energy electrons. We've broken this glucose down all the way now to CO2. We haven't made the water yet. And the energy is now in those high energy electrons. And we're going to take those high energy electrons to the last step, to the ETS, where we'll then make the ATP. Okay? So if I were to ask you on a question, true or false, the Krebs cycle is responsible for making most of the ATP in the cell. False. False, good. Made only one. All right, here's the last part. We're saving the best for last. If you're at the bottom of your notes or the bottom quarter of the page, flip to a new page because you want some space. So this is the ETS, and let's say also oxidative phosphorylation. In a little bit of space, I'll try to use up this board. Here is the outer mitochondrial membrane, and I'm drawing it with the phospholipid bilayer. So this is the outer membrane, the outer mitochondrial membrane. Then we need to leave some space, an intramembrane space, and then we'll drop down and make this convoluted inner mitochondrial membrane. I'll draw the phospholipid bilayer on it also. And then we're going to stick in a series of electron carriers. So they're oftentimes depicted in textbooks as just blobs, either purple or green, depending on the artist. And I'm going to put in 3 of those. And these blobs are representing a series of electron carriers, different molecules, some protein, and this is how they're typically shown or represented. These are the components of the ETS. These are the electron carriers or the electron transport chain represented by these 3 blobs. So what are we well, before we run out of space, let's put in the little mystery pump over here, which we'll label later. But we'll just get it in our diagram so we don't run out of room. We'll come back and talk about that later. This is not part of the ETS. That's going to be making our ATP. So this is the inner mitomembrane. And what's being delivered to the ETS? All of that NADH. So here comes the NADH, bringing with it those 2 high energy electrons. And there's an enzyme that peels off the electrons and the hydrogen. Now, we need to keep track of the electrons. So I'll use a red dashed line for the electrons. So there are our 2 electrons. And they will bounce from one carrier to another in this membrane, causing one to be reduced, another to be oxidized. It's like a bucket brigade, where you pour your water into one next person's bucket, and they pour it into the next person. Your bucket is either full or empty. It's either reduced or oxidized. There's a whole series of electron carriers that flip-flop back and forth, passing the electrons down this chain. Now these are high energy at this point. They're going to flow through the membrane and ultimately leave the ETS as low energy electrons. So here are 2 electrons, and these are low energy. All right, so they gave up some energy. And what these carriers can do, check this out, they're using the energy from the electrons. And what they do is they pump protons or hydrogen ions across this membrane. So the electrons came off of NAD. NAD would then leave this page as NADH would leave as NAD+, right? It has been oxidized. The electrons are passed into the ETS chain. Let me introduce one enzyme, just to kind of give you an idea how enzymes are named. The enzyme that splits off the hydrogen here is conveniently called Nadh reductase, or dehydrogenase. There's the enzyme. It's Nadh dehydrogenase. Does that make sense as far as looking at the substrate for that enzyme, NADH? What did this do? It dehydrogenated it. It pulled off the hydrogens, and then it's left as NAD plus without any hydrogens. So hopefully the name dehydrogenase tips you off that it's removing hydrogens. That's all. Okay, back to the story of the ETS. The electrons flow through the chain, and as they flow through the chain, some of these protein components will pump hydrogen ions across the membrane. And this one will also pump hydrogen ions, or protons, the same thing, across the membrane into that inner membrane space. Okay. That's what the energy from the electrons was used to do. Now, we've talked at some length about chemical gradients. Do you see a proton gradient forming? You should. We're pumping protons from the inner portion of the mitochondria, which is referred to as the matrix, into this intermembrane space. Now these are positively charged. They don't diffuse across the phospholipid membrane easily. So they're trapped out here. Where is there a higher concentration of hydrogen ions? In the matrix or the intermembrane space? Good, in the intermembrane space. What's happening to the pH in the matrix? Just to review. Good, it's going up. More basic. Out here, pH is going down or sitting. Very good. Okay, so we have a high concentration of protons out here. Sometimes referred to as a proton motive force. What do you know about like charges? Do they attract or repel? They repel. So you've got a lot of them out here. They're trying to get away from each other. There's a natural gradient across this membrane, and we let some of those hydrogen ions flow down the gradient back into the cell, and the energy that's released from these protons flowing down their gradient, that energy is used to finally phosphorylate our ADP back to ATP. So we could show that ADP plus P, however you want to show it interacting with this, and forming ATP. And voila, there we go, we get lots of it. Depending on how you do your accounting, the high maximum rate for glucose is about 30 to 34 ATP. 28, to 30, 34, somewhere in that neighborhood. It's not a fixed number. Let's say approximately 28 to 34. That's a lot of ATP for one glucose molecule. What do you want to call this protein? If you want to sort of represent its main activity, let's call it an ATP synthase. Does that make sense? It's synthesizing ATP. Well, gee, there you go. That took less time than I thought, 45 minutes. I knew I was going to race through it. I didn't know I was going to race through it this fast. Few questions, then I'll turn the tape off and we can go back and sort of pick up pieces. It's a lot easier in the book. Oh, this is easier than the book? Oh, okay. Yeah.
So what are the end products of this part?
Yeah, good. Well, what would you say are the end product? The output?
ATP.
Yeah, lots of ATP. That's the big one. That's the important one. And what else came out of this?
NAD plus.
Yeah, and the NAD plus.
Just one of them?
No. There are, well, for glucose, let's run, how many NADs did we make altogether that had to come to the ETS? We had 2 in glycolysis, right? We had 2 out of the prep step, 1 each. There's 1 NADH coming from the conversion of a single pyruvate to a single acetyl-CoA, right? We had 2 of those going on simultaneously. And then how much NADH did we get out of the Krebs cycle? 3. Okay. That was 3 per acetyl group, right? We also got out the FADH2 that was carrying electrons, right? We don't have that up here. We should put that in. And that comes in a little bit lower on the chain. FADH2 comes in here. So let's say FADH2 enters there. So we skipped this first proton pump. So FADH2 isn't going to be able to pump quite as many protons across the membrane. Any other questions? I'm going to turn this off and we'll see what we have. Okay, so backing up to where I left off, I forgot to make the water. I erased this while the camera was off. Here's the inner mitochondrial membrane. Inner mito membrane. The electrons, not all the pieces are shown, but in your notes, we showed the electrons flowing through the ETS. Here were those carriers, right? We had that. There are the 2 low energy electrons. To make water, we have to have H2O. So we bring in 2 protons and we bring in an oxygen. And since oxygen is rarely ever floating around as atomic oxygen, it's molecular, it's bound together. The way we typically show one oxygen is 1/2 of an O2 molecule. And 1/2 of an O2 is just an O. So that goes in. The 2 electrons, the 2 hydrogen ions, the 2 protons, and the half of the oxygen gives H2O. And that is the last piece that we needed for completion of that summary equation. So that's where the water is made at the very end. What is the role here of oxygen? Oxygen serves as the very final electron acceptor. We have these electrons starting out in glucose and being transferred to all sorts of intermediates, wound up on NAD. Those electrons are then passed through the ETS. Who gets them at the end? It's oxygen. Oxygen grabs them up, snags a couple of protons along with it, and we get water. Okay? All right, here's the other 2 little pieces that I'm going to add on that I told you about. What happens in cells? Some cells are able to still make ATP anaerobically. Some of your body cells, your brain cannot do it. But your skeletal muscle cells can make a small amount of ATP anaerobically without oxygen. So all of a sudden there's an emergency, you have to run for your life. Literally. You're running as fast as you can up to Tieton. You cannot supply enough oxygen to keep making ATP at the amounts that are required. So your cells can rely on glycolysis. Glycolysis itself is anaerobic. There's no oxygen that was required for glycolysis. So the step of glycolysis is anaerobic. You need the oxygen in the ETS to make all that ATP. So let's make some more, let's run through glycolysis again. It's an anaerobic process. So we'll start out with glucose. We'll just do that summary reaction. Glucose to 2-pyruvate. It's exactly like we did before. We're just writing out glycolysis. We had 2 ATP going in, 4 ATP coming out. That looks familiar. Now here is the important piece. NAD plus oxidized went in and came out reduced as NADH. Now, in order to keep glycolysis going, what do you need? What are the inputs to keep glycolysis going? You need a little ATP, okay. You need glucose, but you need this. You need NAD to pick up the electrons. If you don't have this, you can't do glycolysis, and you can't get out your little bit of ATP. So, where does this normally go? To the ETS. And those electrons that are here, after they flow through the ETS, where do these electrons wind up? On oxygen as water. Oxygen was the final electron acceptor. Yeah? So if we don't have enough oxygen to accept these electrons, this whole thing backs up and it won't proceed. So all we have to do is drop off the electrons, right? If we could unload the bus, kick off the 2 electrons, Then we're back up to here. And we can keep running glycolysis. Yeah? So where are we going to send these two electrons? Well, we can put them back onto pyruvate. And when we do that, we make lactate, or lactic acid. So we could represent when we add 2 electrons here to pyruvate, what are we doing to pyruvate? Reducing it or oxidizing it? Yeah. So you could say that we've reduced pyruvate to lactate. There are those 2 electrons. They're back on the pyruvate. And what we did was we regenerated the NAD. That's the whole purpose of lactic acid fermentation. Is to regenerate that NAD. And that's the purpose in this story. Okay? For lactic acid fermentation. And you probably have heard, you exercise your muscles really hard, they hurt later. One of the ideas is that it's because of the lactic acid buildup in the muscles. There's a lot of energy in lactic acid. That gets picked back up, it's eventually flushed out, sent to the liver, converted back to pyruvate, and away we go again. So that takes some time. Lactic acid fermentation. Are you going to be able to draw it and explain its significance in the task of regenerating NAD? The last piece, I know your brains are full. This has been a busy day. 70 minutes of this stuff is not easy to sit through. You all are doing pretty good. Well, here, maybe this will be more familiar to some. Alcohol fermentation. Maybe you're dreaming about this right about now. Hopefully not too bad. We start out again with glucose and go to pyruvate. Same story. 2 ATP in, 4 ATP out, NAD plus in, NAD, H out. Certain organisms like yeast, not us, we can't do this, but yeast can, they will convert the pyruvate, they can do this anaerobically. Anaerobic, you lock little yeast in a bottle with sugar and they can turn it into ethanol. That ultimately, through a series of steps, gets converted to ethanol, which is the type of alcohol in beverages. And ethanol is a 2-carbon alcohol. Pyruvate had how many carbons? 3. So we lost one as a CO2. And that's what makes the beer bubbly, is the carbonation, the CO2. But otherwise, everything else is exactly the same. Pyruvate gets reduced to an intermediate, which ultimately gets converted to ethanol, but we wind up regenerating the NAD anaerobically. The byproducts, ethanol and CO2. That's also, well, okay. That's it. We're out of time. Probably out of tape. Brooke? Yeah. No, sorry. used, they're attached to the pyruvate to reduce pyruvate. So the up part of that green arrow is without electrons.